Thank you. And thank you to the Academy for my election. It's a great honour. I'd also like to thank all my colleagues, collaborators, CSIRO for providing a stimulating place in which to work, the two universities where I'm adjunct, and also to the GRDC, the farmer organisation, who has supported my work. I'd also like to thank Australian farmers who have been my close collaborators throughout my career, and who many of you may not know lead the world in the adoption of conservation farming systems, as you see here, and have led the transition from the type of farming system you see on your left to your right. And I want to talk to you about that transition. So if we take the national average wheat yield as an indicator of productivity and sustainability of the system, we know that science along the way has had a big role in supporting that trend. You can see that after an initial period of degradation, it took us some time to recover, and many of the fellows in this academy have been involved in that process. What I wanted to focus on is this last period, this last 20 to 30 years in which I've spent my career, and to talk to you about a few of the scientific applications and some that my team have been involved with to drive a period where we doubled wheat yields and arguably have now the most sustainable system we've ever had. The President said at the start that it takes multidisciplinary teams, and that has certainly been the case for me. As an agronomist, I sit in the middle of those intersecting circles, and I've had the privilege to work with fundamental scientists down at a cellular and plant scale at one end, and to modellers and industry people at the other end, so that we can have impact. And I want to talk about a couple of examples where I hope to show you that interaction in action. So the Australian dryland mixed farming system that we focus on in my team is shown there in that yellow band. It's a very large area and has substantial economic impact on the country. The two examples I want to talk about are the introduction of more diversity into that farming system, with the introduction in this case of canola, edible oilseed rape, a plant that was, as you can see there, area has increased enormously in the last 20 to 30 years. And I'll come back to that period of the millennium drought a little later. And the other one I wanted to talk to you was about the introduction of no-till farming, that is, cultivating plants, growing plants without any disturbance of the soil by sowing seeds directly into undisturbed residue of last year's crop. This has been, as you can see again, adopted widely and quickly by Australian farmers. So just going to canola. Canola is an exceptional rotation crop for wheat, and you can see there in the picture, picture tells 1,000 words. When farmers saw wheat crops growing after canola compared to what they had been used to, of course, it was easy for them to see those benefits. The graph shows the yield improvement to wheat growing after canola, all the points being above that line showing that there's substantial improvements in yield of wheat when canola is used as a rotation crop. Not only that, what was more intriguing was that canola appeared to be more effective, and other brassicas like mustard seemed to be more effective than some of the other rotation crops we had in the system at the time, and it was intriguing for us to understand why that should be. One of the unique features of brassica crops is the fact that they contain these sulphur-based compounds called glucosinolates. There's hundreds of different kinds of glucosinolates based on the type of R side chain there. And although you don't know it, many of you are familiar with this reaction. When you put a mustard seed in your mouth and you chew it, the hot burst in your mouth is an isothiocyanate, a volatile compound being released when the glucosinolate is hydrolysed. So it made us wonder whether these chemicals may have something to do with the special properties of canola, given that isothiocyanates are actually produced as soil fumigants and were being sold as soil fumigants. So we did some research. I guess we focused on the roots, because that was really the only part of the canola plant that was in contact with the soil, and we did discover that brassicas do concentrate a specific glucosinolate, an aromatic compound, in their roots. It turns out to be quite toxic to some of the major fungal pathogens that were in the soil and attacking wheat crops, the take-all fungus, the black colour inside that wheat root being an example. And indeed, when we identified canola varieties which had higher levels of this compound, and we grew them in soil preceding wheat, we could substantially reduce the level of this disease. Now, as happens in science, the application of this idea, this biofumigation concept, wasn't necessarily taken up in this context, but where it was taken up was in horticulture. At this period, you'll recall methyl bromide, which was a fumigant widely used in the horticultural industry, was being phased out because of its impact on the ozone layer. And so we wondered whether or not we could utilise some of the naturally produced isothiocyanates in brassica plants as green manures and use them in horticulture to control some of the diseases which formerly had been controlled by methyl bromide. After a lot of investigations of what types of compounds were there, how toxic they were to various pathogens, and a lot of chemistry and biochemistry in the soil, we were indeed able to demonstrate that on particularly sandy soils where the compounds weren't tied up in organic matter, we could have quite substantial impacts on diseases like bacterial wilt in solanaceous crops, and have better results with brassica green manures than even legumes. And in places like Washington State in the US, they've now adopted this technology. They now have completely replaced the use of synthetic fumigants with mustard, in this case mustard green manure crops, with not only control of disease but other benefits to the soil, savings in energy and in CO2 production. And now there's 25 different countries utilising this sort of technology to assist to replace these synthetic fumigants, which are slowly being banned from use. Coming back to the Australian dryland situation, and I recall the drought I showed you, during that drought canola started to really slip out of the system again after getting it adopted and widely used because of its drought sensitivity. And in order to arrest this decline, we needed to find a way to make canola less risky for farmers so they would keep it in the system to get those rotational benefits. The idea we came up with was to see if we could get a second income off the canola by developing crops that could be grazed in the winter while they're leafy and still be grown on to produce grain. That is a dual purpose crop. And again, by collaborating widely with breeders, physiologists to understand the response of canola to defoliation, animal scientists to understand their value as a feed, simulation modelers to help work out where this might have application, we were able to successfully develop these crops, and they're now adopted by farmers in every southern state in Australia. Moving now from canola to no-till farming, it's not hard to imagine why not having to go round and round fields and cultivate the soil would save soil, labour, fuel, machinery, and improve soil conditions. And certainly that's what farmers saw. But interestingly, they were not necessarily seeing their crop yields increase as a result, which was quite disappointing. The soil seemed to be improving, but in long-term studies, the crop yield was not responding. We wondered why that was. Again, there are particular diseases that like uncultivated soil. We knew about those, we could deal with those, but even with those, it just seemed that plants growing in uncultivated soil grew slowly, almost like a bonsai effect. And initially we were focused on the fact that this uncultivated soil was hard and potentially limiting crop growth, until we fumigated the soil and found that that completely alleviated the problem. So did we have a physical or a biological problem here? This was solved with some elegant work by Michelle Watt, a postdoctoral fellow, who had a really close look, and I mean a really close look at roots growing in the soil, and showed that roots growing in no-till soil were slowed down by the hard soil. And it wasn't so much that slowdown itself that was causing the problem, but the fact that when you slow a root down, bacteria in the soil build up on the exudates coming from that root, seen here with a fluorescent technique showing the bacteria on the root tip. And some of these bacteria, when they get to a certain population, will exude inhibitory compounds, mostly hormonal compounds, which will slow down wheat growth. So this was an interaction between a biological and a physical condition in the soil. To overcome it, the farmers were ahead of us. Sow the crop early when the soil is warm and the roots already grow quickly because of the temperature. Disturb the soil below the seed rather than sideways, and give the root a quick exit out of that hard soil. Or working with our breeders, we were able to demonstrate that you can find wheat varieties which have inherently fast roots and can overcome some of this issue using different varieties, more vigorous varieties. So this is the farming system that we came from: continuous wheat with sheep grazing weedy fallows, burn and cultivate the residue, sow the crop after you get enough rain in May, and you'll get a wheat yield about 1.6 tonnes per hectare, which I showed you was the national average. I've talked to you about the fact that a better rotation can help, and certainly it can raise the yield a little. I've talked to you about the fact that once we overcame the issues, no-till farming can not only protect the soil but also give us some yield improvements. But these are really disappointing when you look at them and when we know the physiological potential. So what was missing? In this case, one thing that was missing was that sheep grazing weedy fallows, those weeds are actually utilising water, which is then unavailable to the crop. And so if we control those summer weeds rather than let the sheep graze them, we could get a much larger individual impact, but it still hasn't taken us a long way. When we started putting these together in a system, we realised that we could store more water, therefore being able to sow the crop earlier. We also found varieties of wheat that had longer coleoptiles to allow us to sow them deeper into that stored moisture. And when we put that whole package of innovations together, we could take the wheat yield from 1.6 to 4.5, which was the physiological potential. So I think you can see, and Graham talked earlier about the efficiency of water being used by plants. We have to have the system around those plants that make the water available, keep it there and allow them to capture that benefit. And I'll finish by just saying the revolution continues. We have a lot of new genetic techniques, better crop varieties, with all sorts of new technologies to make more rapid the kinds of traits and plants that we can use in agriculture. And I think we may hear from Alex McBrackney after lunch. We also have the digital agriculture revolution providing just unprecedented precision with the way we can deploy these plants in the field. So with that, I'll thank you for listening. Take questions.
Thank you for being precisely on time. Any questions for John? Yes, we've got a question over there. I'll encourage you to be monosyllabic in your answer.
How much did you have to change the breeding of the wheat to suit this, what sounds like a completely new system?
The major change was to change the phenology of the wheat, to allow us to sow earlier but have the wheat flower at the same time, because wheat is squeezed between the last frost, which will kill the flowers, and heat, which will prevent the crop from filling the grain. So one of the main changes was changing the phenology, so the way the crop responded to temperature and day length, so it flowered at the same time. And that long coleoptile also allowed us to sow the crop deeply into that stored moisture. The kind of changes that are predicted under climate change that are coming, that whole system I've just described is very well adapted to what's happening. And that's a lack of autumn rainfall, sustained summer rainfall and variable spring rainfall. Those changes that system I mentioned is very well attuned to adapting to that predicted change in the climate.
Okay, I think we should call questions to a halt there. Sorry about that. Thank you very much, John.